Hello friends, today I will be discussing about the Operon concept. So this is a gene regulation mechanism. So let me first define what is an operon. An operon is a part of genetic material. This is the DNA, the part of genetic material which acts as a single regulated unit consisting of one or more structural genes, one inhibitory gene, and one promoter gene and an operator gene. These operon systems are common in prokaryotes, and the operon can also be defined as a group of coordinated genes which function together and regulate a metabolic pathway as a unit. The first operon system was discovered as the lactose operon. Lactose operon. It was discovered by Jacob and Monod in 1961 in E. coli bacterial cell. Okay. So later on several other operon systems were also discovered. So let's have a discussion on lactose operon. Lactose operon. The lactose operon consists of four types of gene. So the first, number one is the inhibitory gene called I gene or regulatory gene. Its product is a repressor protein. When the gene express, it produces the repressor protein. The repressor protein has the affinity for the operator gene and it also interact with the inducer molecule. Okay. The second is the promoter gene. Promoter gene, it provides binding site. To RNA polymerase. You know the enzyme RNA polymerase that helps in transcription. So here, this is the operon system. So this is the promoter gene, and this site or this gene provides binding site to RNA polymerase. RNA polymerase attached to this site. So the third is the operator gene. The operator gene receives the product of inhibitor gene. Means the operator gene receives the product of inhibitor gene, that is the repressor protein. When it is covered over, this operator gene is covered over by this repressor protein, then the functioning of the operon is stopped. Means the operon is switched off or turned off. And when this operator's gene is not covered over by this repressor protein, the operon is switched on or turned on. So thus it allows the functioning of the operon when it is not covered over by the repressor protein. Okay. The fourth is the structural gene. So here in lactose operon there are three structural genes: gene A, gene Y, that is Lac A gene, Lac Y gene, and Lac Z gene. So the first is the Lac A gene. It codes for an enzyme trans-acetylase. You know the gene, when gene undergoes expression, it produces functional proteins or enzymes. So this gene codes for trans-acetylase enzyme. So this enzyme transfer acetyl group to beta galactoside. So in biochemistry, the beta galactoside is the lactose, or it is called a lactose. Then the second structural gene is the Lac Y gene. This gene codes for an enzyme that is permease. This is a transmembrane channel protein. Transmembrane channel protein. So this permease enzyme allows the entry of lactose into the bacterial cell. Means the enzyme allows the lactose, the disaccharide lactose, into the bacterial cell. See, that is the Lac Z gene. This gene codes for the enzyme beta galactosidase. Beta galactosidase. So this enzyme hydrolyze lactose into glucose and galactose. Glucose and galactose. Okay. So the lactose is a disaccharide. The beta galactosidase enzyme break down the lactose into glucose and galactose. Okay, these are the three structural genes. So in lactose operon, the four types of genes, the inhibitory gene or regulatory gene, its product is the repressor protein, and the promoter gene, it provides binding site to RNA polymerase, and the operator gene, it allows the functioning of the operon when it is not covered over by the repressor protein. When it is free, it allows the RNA polymerase. So that when it is free, the RNA polymerase access over the structural gene, so that the operon is turned on or switched on. So let's have a discussion when the operon is switched off or switched on. So first we'll discuss operon switched off. Operon switched off or turn off. Turn off. Naturally, this operon system, the lactose operon, naturally remains switched off. When there is no lactose, suppose lactose is absent in the bacterial cell, lactose is absent. If there is no lactose, the three enzymes, that is, so we have already discussed the three enzymes, that is gene A, structural gene Y, and gene Z. The gene A expresses to form, that is the enzyme transacetylase, and it is a permease, and this third one is the beta galactosidase. Okay, so it is permease, this transacetylase. Okay, if lactose is absent, these three enzymes is not required for the cell. So what will do these three enzymes if there is no lactose? So the genes should be switched off or turned off. So it happens so. So this is the inhibitor gene, and this is the promoter gene, and it's the operator gene. Okay. So the inhibitor gene always expresses in the cell. It produces the repressor protein. The repressor protein. When there is no lactose in the cell, lactose here absent. Lactose is absent. Okay. There is no lactose. No lactose in the cell. Okay. In absence of lactose, this repressor protein binds to this operator site, so that the RNA polymerase have no access over the structural gene. So there is no transcription and translation, and these three enzymes are not synthesized because lactose is absent. So there is no need of these three enzymes. So the gene remains, or the operon is switched off. Switched off. That is switched off condition. This is a kind of gene regulation mechanism. Now these enzymes are not needed by the cell, so the operon remains switched off. Let's discuss about the operon switched on. Switched on or turn on. Turn on. Suppose there is lactose inside the cell, and we provide lactose in the growth media so that the bacteria get the lactose. There is lactose in the growth media, and the bacteria get this lactose. Now these three enzymes are required for the breakdown of lactose, gene A, Y, Z. These three genes must be expressed so that these three enzymes are produced, which break down this lactose so that the bacterial cell get energy. So in presence of lactose, so lactose here called inducer molecule. When there is lactose in the cell, the inhibitor gene, that is continuously expressed, that it is the repressor protein. Its product is the repressor protein. In presence of lactose, the lactose interacts with the inducer molecule. So So this is the lactose. Lactose interacts with the inducer molecule, means that becomes inactive. So lactose or inducer plus repressor equal to inactive repressor, because the lactose changes the conformational changes of this repressor protein so that it cannot bind to this operator site. Now the operator regions 3. Okay, so here the lactose is called the inducer molecule, and this lactose operon is called inducible system. So here the question, why it is called inducible system, lactose operon is called inducible system, because lactose itself induces the expression of this gene. In presence of lactose, the gene is expressed, so it is called an inducible system of operon. So here, when lactose interacts with the repressor protein, the repressor becomes inactive. Now the operator regions remain free, and the RNA polymerase have access over the structural gene, so that there is transcription and a mRNA molecule is produced, and this mRNA is called polycystonic. Why it is called polycystonic? Because it carries the information of these 3 cystine. So this structural gene, otherwise called cystron, here there is 3 cystine, and 3 cystine expresses to form a single mRNA molecule. So it is the prokaryotic genes, prokaryotic mRNA are called polycystonic, but in eukaryotes we have monocystonic mRNA. And the prokaryotes are polycystonic mRNA because the mRNA carries information of these 3 cystine in this lactose operon. Okay. So after transcription, a polycystonic mRNA is produced, and then translation occurs. These 3 enzymes are produced. First enzyme, second enzyme, third enzyme, that is the gene A codes for transacetylase, and gene Y codes for permease, and gene Z codes for beta galactosidase. Now these 3 enzymes act upon lactose, and lactose is broken down to form glucose and galactose. Further breakdown of glucose and galactose produces energy, so that the bacterial cell gets energy. So it is a type of gene regulation mechanism, and this lactose operon is called the inducible operon system. I hope this video will help you in understanding the lactose operon. Okay, thanks.